So hi everyone, I'm from group 322. My name is Palak. So my today's topic is auto-oscillation and glycolysis and photosynthesis. So auto-oscillation and glycolysis refers to phenomena where metabolic pathway exhibits oscillatory behavior and concentration of certain metabolites. This can occur due to non-linear dynamics of enzymatic reaction involved in glycolysis, particularly under certain conditions such as varying substrate concentrations, enzyme activities, or feedback mechanisms. In glycolysis, glucose is broken down into pyrite, producing ATP NADH in process. The pathway involves several key enzymes that can be subject to regulation such as phosphofructokinase and pyrite kinase. The activity of these enzymes can be influenced by various factors including ostratic regulation, feedback inhibition, coupled reactions, external stimuli. Key concepts of auto-oxidation and glycolysis. Nonlinear dynamics. Enzymes involved in glycolysis can exhibit nonlinear kinetics, meaning that their reaction rates don't change linearly with substrate concentration. This can lead to feedback loops that contribute to oscillatory behavior. Key enzymes of regulation. Phosphofructokinase, major regulatory enzyme in glycolysis, and it is also strictly inhibited by high levels of ATP and citric, while being activated by AMP and fructose 2,6-biphosphate. Changes in concentrations of metabolites can lead to oscillation in fructokinase activity. Pyrite kinase enzyme also plays a critical role in regulatory final steps of glycolysis and can be influenced by various metabolites, contributing to feedback loops. Feedback mechanism. Accumulation of products can inhibit upstream enzymes, leading to reduction in glycolytic flux. Conversely, if ATP levels drop, PFP activate may increase, leading to reassurance in glycolytic activity. The interplay can create oscillations. Coupled pathways. Glycolysis doesn't operate in isolation. It is coupled with other metabolic pathways such as citric acid cycle, oxidative phosphorylation, fermentation. These interactions between these pathways can influence dynamics of glycolysis, potentially leading to oscillatory behavior. Experimental observations. Auto-oxidation has been observed in various experimental setups, particularly under conditions where substrates or metabolic intermediates are varied. Biological significance. Oscillation in metabolic pathways like glycolysis can have important indications for cellular function. They may help cells adapt to changing environmental conditions, optimize energy production. Auto-oscillation and photosynthesis. It refers to periodic fluctuations in rates of photosynthetic process such as production of oxygen or fixation of carbon dioxide that can occur under certain conditions. This phenomena can be influenced by environmental conditions, dynamics of chloroplast metabolism, and interactions between biochemical pathways involved in photosynthesis. Key concepts. Overview of photosynthesis. Process by which plants, algae, and some bacteria convert light into chemical energy, primarily through conversion of CO2 and HO2 into glucose and O2 using sunlight. There are two types of sunlight influenced photosynthesis. So one is light dependent reactions. These reactions occur in the thylakoid membranes of chloroplast and involve the observation of light by chlorophyll, leading to the generation of ATP and NADH. The rate of these reactions can fluctuate based on light intensity and quality. Second one is light independent reactions, also known as Calvin cycle. The Calvin cycle occur in the stroma of chloroplast and use ATP and ADPH produced in the light dependent reactions to fix carbon dioxide into organic molecules. The rate of Calvin cycle can also show oscillatory behavior and based on the availability of substrates and energy carriers. Nonlinear dynamics. The interactions between various components of the photosynthetic machinery can exhibit non-linear dynamics. For example, feedback mechanism involving metabolites like ribose biphosphate, which is a key substrate in the Calvin cycle, can lead to oscillation at its concentration and affect overall photosynthetic efficiency. Environmental factors. Factors such as changes in light intensity, temperature, CO2 concentration, and water availability can influence the rates of photosynthesis. Rapid changes in these environmental conditions can induce oscillations as photosynthetic apparatus adjust to optimize energy capture and carbon fixation. Metabolic feedback loops. The production and consumption of metabolites with chloroplast can create feedback loops that contribute to oscillatory behavior. For instance, an accumulation of certain intermediates may exhibit or activate specific enzymes involved in photosynthetic, leading to periodic changes in metabolic flux. Experimental observations. Researchers have observed oscillatory chlorophyll, fluorescence, oxygen evolution, and carbon fixation rates in various plant species under controlled experimental conditions. This oscillation can be influenced by factors such as nutrition availability and the presence of stressors. Biological significance may play a role in optimizing energy use, carbon fixation under fluctuating environmental conditions. Talking about the conclusion, auto-oscillation in photosynthesis is a complex phenomenon that arises from the intricate interplay between light capture, metabolic processes, and environmental factors. Understanding these oscillations can provide insights into how plants adapt to changing conditions and optimize their photosynthetic efficiency. Further research in this area has implications for